Landontopia, our eco-column project.
Brief description, ecological. In our Landontopia ecological system, we had 2 aquatic plants, 1 radish, 1 terrestrial plant, 1 fish, 1 cricket. The day after we started, both our fish and cricket died. We believe that the cricket was trying to escape, as we found it trapped in the tank. We are not exactly sure why our fish died, but we suspect that it either ran out of oxygen to breathe or it did not have a sustainable source of food. A couple days after our fish had died, it started to decay and smell very bad. A cloudy membrane surrounded the fish and pieces of it started breaking off and floating around. Our radish was starting to grow during this time, but however we could not find it when we started looking for it on the last day. We are not sure what exactly happened to it, and we hope that we will find out if we open our eco-column.
Brief description of the variables. We measured the dissolved oxygen levels in the aquatic chamber, the pH balance in the aquatic chamber, and the oxygen levels in the terrestrial chamber. The dissolved oxygen levels fluctuated throughout the duration of our experiment. The initial level was 3 milligrams per liter, and it rose to around 7 milligrams per liter within 5 days and gradually fell to around 4 milligrams per liter in the duration of 9 days. The pH balance stayed pretty much constant at 7 to 7.3 throughout the entire experiment. The oxygen levels in the terrestrial chamber decreased slowly during the experiment. Throughout the 14 days, the oxygen levels slowly decreased from 20.63% to 19.67%, with a small jump up to 20.24% on the 11th day.
These graphs are the graphs we use to represent the amount that each of the three things that we measured went by. Like Sam said earlier, just said before, dissolved oxygen went at first from 3 milligrams per liter all the way up to 6.7 at its highest, and going down a little bit by about 0.7, and then down to 3.6 at the 27th, and up in the end of our experiment to 4.2. pH balance went all the way from 6.9 to a maximum of 7.3, going down again up to 7 and up to 7.13. These pictures were taken before and after from the experiment, since from the beginning, from how our experiment looked when it was all full of life and before our fish died and stuff. And the after picture is one we took a couple of days ago, and how it looks very different from what we began with.
So after looking at our results from our initial experiment, we came up with the following question. How do fish live in the water supply affect the nitrate levels in the water compared to when there are no fish?
Study design. First, place the fish in one of the plants in one bottle and the other plant in the other. Take an initial reading of the nitrate level in both columns. Every day, take a measurement of the nitrate level in both systems. Take notes of how the organisms appear to be varying in both systems, such as the color of the leaves or the health of the fish. For example, is the fish floating around or is it swimming? Is the fish alive or dead? Repeat this process for 2 weeks and conclude results.
Our hypothesis, which is looking at the health of our plants in our ecosystem to decay fish, we predict that the fish being present in the water will help decrease the nitrate levels in the eco-columns.
Our expected results from our experiment is that the nitrate in the eco column with the fish would be lower than the eco column without the fish. We believe that the fish being present will decrease the nitrate level because the fish will affect and be affected by nitrates present in the water.
Contradictory results. There are a couple situations in which the results we receive will contradict our hypothesis. For example, if the nitrate levels don't change even with the fish in the eco column, then we would conclude that the fish did not affect the nitrate levels, or if the nitrate levels are higher in the eco column with the fish, we can conclude that the fish increased the nitrate levels rather than decreased them. Also, if the levels of nitrates increase or decrease in both eco columns, it means that the fish has nothing to do with the nitrate levels, and there is another cause to the change.